PCR, or you're going to do a microarray analysis. But they're also very useful for mapping genes. And the reason is, you have over 1 million SNPs between one individual and the next individual. So any two individuals, if you compare their genome sequences, they will be different for about 1 million different SNPs along the chromosome. And so you can follow a pedigree of a father, a mother, and their children by following the SNPs and how they are migrating. So instead of analyzing all 3 billion base pairs, you analyze just the 1 million SNPs and determine how a fragment of a chromosome is migrating from one parent to the child to the grandchild. And so you can use SNPs to map genes as well. So SNPs occur every 1,000 base pairs. Classical Mendelian, just imagine, you have 3 billion base pairs of DNA. You have 200 markers, right? So each marker is spaced very far apart. I can't do the math at this time. 3 billion divided by 200, you do the math, you'll figure out approximately on average how far apart each of those markers are located. If you use RFLPs, there are about 7,000 RFLPs that we have mapped in the human genome across the 3 billion base pairs. So yes, you get a much better picture when you have 7,000 markers compared to 200 classical Mendelian markers. But look at the SNPs. You have 1 million SNPs across 3 billion base pairs. So you can add, the beauty is, you can analyze all 1 million SNPs at the same time, simultaneously, using microarrays. So you can get a picture of the entire genome and determine where a mutant gene is segregating with which SNP across a pedigree. So this is the power that we are now dealing with. Okay, so humans are 99% identical. And it's that tiny difference, a fragment of that tiny difference is basically the SNPs, and that's what we're going to use to study how one individual differs from another. So how do SNPs arise? Let's imagine this is the parent sequence. That's the F1 generation, and they have 2 children. This parent has 2 children. Imagine that. During the process of producing gametes, right? The ovaries are in the sperm, in the testes. During the production of egg and sperm, mutations may form, and those mutations are then passed on. If that mutant sperm or the mutant egg gets fertilized, it gets passed on to the next generation. Now it will get passed on forever. So imagine here is a normal sequence. Here is one of the children, and you notice this child's sequence is identical to the parent sequence in these locations. No difference. It's exactly the same. ACTG, ACTG, GAC, whatever. But in this child here, second child, there is a change. This C over here is converted to a G. That's a SNP. That happens spontaneously during the production of a gamete in the father or the mother. That egg or sperm with that change was fertilized and became a child. Now that child will have it in its genome and pass it on to his or her children and so on. Now this F1 may have a child, and now you may have a spontaneous change over here. Alternatively, this F1 child may have a second child. This G is fixed, and now there's a spontaneous change here. And so you can study the relationship. You can say, this individual is very close to this individual. This individual is close. This individual is very close to this individual, slightly close to this individual, based on the number of SNPs that are similar and the SNPs. And so you can adjust and determine relationships between individuals in a pedigree using these sequences and these SNPs. So you compare the two F2s, this F2 with this F2, and you can see that there are 3 differences that are repeated. And so on. So you can basically determine the relationship of these different individuals. And that's how you determine relationships. So each of your 10 to the 13 cells in the human body receives a huge number of lesions. You've seen this slide before, every day. When these mutations are not repaired, they get fixed in the cells. Now, if a mutation is fixed in germ cells, and that germ cell gets fertilized, you will get progeny that will have that change in the sequence. And there are approximately 70 new SNPs being introduced in the progeny in every generation. Between the parent and the new progeny, there are about 70 different changes. These are spontaneous changes. So I want to show you this slide just to give you a relationship of the terms we've been using. The relationship between a SNP, an RFLP, and a point mutation. So imagine there are 3 genes, 1, 2, and 3. And we're just going to focus on one restriction site, GAATTC. That's the recognition site for EcoRF. So you have a site over here, you have a site here, 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 and here. So this restriction site for EcoRF is within this gene. These two are not within the gene. This one is also not within the gene, and likewise. So those are restriction sites, nothing else at this point. Now, imagine a situation. This restriction, this is a different individual. This restriction site remains the same. No change. This restriction site in this individual disappears. Instead of being GAATTC, you are GAGTTC. This is an RFLP. Correct? Because you have a restriction site that's disappearing in one individual but present in the other individual. So you're going to have a change in the restriction fragments that you analyze with the So that's an RFLP. This restriction site remains the same, GAATTC. This restriction site remains the same. And here is another restriction site that's changed, GAATTC, changes to a GACTTC. So again, there is an RFLP. But is that the only thing that you notice? You may have a SNP, a change in the nucleotide sequence that's located here. It's not within the restriction sites. That's a SNP, single nucleotide change. So, but it's not within a restriction site, so it's called a SNP. It's not called an RFLP. This sequence change that you saw in this restriction site is an RFLP, but it's also a SNP because you have a change in a single nucleotide. So this is a SNP as well as an RFLP. You can have a SNP over here within a gene. It's not within a restriction fragment or restriction site, so it's a SNP, but it's also a point mutation because it's within the body of this gene and it's mutating the gene. So that's a point mutation. It could be a frame shift mutation, a nonsense mutation, something, but it's a point mutation. Sorry, not a frame shift, a missense mutation. This mutation within the body of the gene that changed this EcoR1 site to a non-EcoR1 site is an RFLP. It's also a SNP because it's a single nucleotide change, and it's also a point mutation because it happens within the body of the gene. So you can see the relationship. All single nucleotide changes are SNPs. Some of them are RFLPs. Some of them are mutants, point mutations. Some RFLPs are point mutations, some RFLPs are not point mutations. But they are outside of the gene. So terminology depends on where the change takes place. And that's how you refer to it. Okay, so there are 2 types of SNPs. You can get SNPs within the coding region of a gene. You can get SNPs outside of the coding region of a gene. Within the coding region of a gene, you again have 2 types of SNPs. Synonymous SNPs, these are basically silent mutations. They don't change the sequence of the protein, so they don't have a phenotype. but they are changing the sequence of the DNA. Because of the redundancy of the genetic code, you can have silent mutations, and those are known as synonymous genes if they're in the body of the gene. And then you have the non-synonymous genes. These are the ones that will lead to a mutation within the coding region. Now, there is a famous Alzheimer's SNP. I've talked about this before, the E4 SNP. These are mutations. There are 3 different sequence variations within the gene for a particular protein that's involved in Alzheimer's, or apolipoprotein E4. And within this gene, there are 3 different alleles, E2, E3, and E4. These are single nucleotide changes in the sequence of the gene. And it's this E4 gene that correlates very strongly with individuals that have Alzheimer's. And so that's a point mutation and a SNP. It's not an RFLP, it's just a point mutation. Okay. And then of course you have SNPs that are not within genes but outside of genes. These are intergenic SNPs. And it's usually the intergenic SNPs that we use to map genes. Because these are mutations or changes in sequence, they're not mutations, they're polymorphisms that don't affect any phenotype but allow you to study genes in their neighborhood and how they are segregating together because of the linkage. So what we're going to do is we're going to compare SNP patterns of individuals who are affected and individuals who are not affected, and then we'll catalog all of the changes across the entire genome, and hopefully we'll be able to identify the location of a mutant phenotype based on that correlation. So we're going to do something very similar to what we were doing with RFLPs and pedigrees. Except instead of using RFLP patterns, we're going to look at SNP genes, single nucleotide genes. That's the only difference that we're going to do. So how do we identify SNPs? How do we study SNPs? 2 ways, PCR and microarrays. So, we can use PCR. Now remember, PCR is useful because there's no cloning necessity of that region. Once a region has been sequenced in one individual, you can use that information to study that same region in other individuals. So, you don't have to clone the entire region once it's been sequenced in one individual. You need only very tiny amounts of genomic DNA. And you get many, many copies of it, and you can analyze those copies by amplifying. So, quick overview of PCR, one more time for you guys. Here is the region you want to amplify, with some flanking sequences on either side. You design primers, one primer outside of this red region that you want to amplify, going 5' to 3' left to right. One primer on this side, going right to left 5'. Tiny little fragments, either 5 nucleotides, 10 nucleotides, 20 nucleotides. In reality, it's usually 20 nucleotide primers that you use, but 20 nucleotides gets long for you guys to analyze, so we can get 6 and 7 nucleotide sequences. So PCR is basically you're going to denature the DNA. The primers are going to be new. Then you add the enzyme and you synthesize the second strand of DNA. And you repeat this entire process 30 times. And when you do this experiment, you will get a fragment of DNA in a particular size. And you can run it on a gel and it will appear. Right? That's PCR. Simple method. Okay. So how do we study a SNP? So what you have to do is basically you have to identify a SNP first. Let's imagine I am sequencing the genomes of 3 different individuals in this class. And based on that, or 4 different individuals in the class. Let's make it slightly better in terms of statistics. 4 individuals. I sequence the genomes of 4 individuals in this class, and I find 2 individuals have one particular sequence at position, 2 other individuals have a totally different sequence at that position. So I say, okay, that's a SNP. There's a single nucleotide polymorphism at that one spot. Now I'm going to design my primers for PCR using that sequence information. So let's imagine there is individual 1. I've sequenced them, and here is the sequence. And here is individual 2, and here is their sequence. Notice instead of an A over here, individual 2 has a T. So that's a SNP. Now, once I know this information, based on sequencing of a small group of individuals, I can now use this information to analyze all of you using PCR. At that one position, I can determine there now which allele you have. Is it the A allele or the T allele? And the way you do this is you design primers. So imagine this is one sequence with the A allele, and here is the T allele. That's a little point change. When I make a primer, that's going to hybridize exactly perfectly with this sequence. So it will anneal to it and run through, correct? Because of the PCR reaction. This primer, if I were to anneal it like this, it's going to have a base in the middle that's not going to anneal. It's not going to form a base pair because of the change. The sequence of the primer isn't going to match the A, it's not going to match the T. Now, when I start the reaction by raising the temperature, this primer, because it's perfectly matched, will stay bound during the PCR reaction. This primer, on the other hand, will fall off. It won't stay bound. And if it doesn't stay bound to the sequence, you can't get a PCR reaction to work. And so what happens is, when you have this sequence, you will get a product. Or when you have this sequence here, with this primer, you get no product from PCR gel. So you'll be able to determine that this individual has a change of a single nucleotide, what this individual has. Isolate DNA from individuals 1 and 2, and test them under conditions that only allow perfect matches of the little primers to the individual. So if you cannot anneal, you cannot get a PCR product, and you get a blank spot on the chain. And that's an indication that you have other information. So, mutants cannot be amplified, the wild type situation is amplified. Here is the sequence, primers. These are, say, the 2 primers. These primers are perfect matches for the wild type individual, and you will get a product like this. But now let's imagine now here we have a primer that doesn't match perfectly. Maybe I messed up. Here. That's okay. Same primers, but now you have an individual with a SNP. There's your SNP. And now the primers don't match. This primer doesn't match because of the change in the sequence. And because of that, you don't get an amplification. This primer cannot hybridize, it cannot extend the DNA, you don't get an amplified product. You basically can't see what happens. That's what. So PCR allows you to do one SNP to be tested at a time. You can analyze a single SNP in a population of individuals, one at a time. So we can analyze this one SNP. Then you go on to the next SNP, 1,000 base pairs, you analyze that. The third one, you analyze that. The fourth one, you analyze that. So in effect, you can do it, but you can only do one SNP at a time. It's a time-consuming method. It's not very efficient. It's very sensitive, but it's time-consuming. On the other hand, you can use microarrays to analyze a whole slew of different SNPs all at the same time. Remember, what are microarrays? They're little glass slides with single-stranded molecules of DNA stuck to them, correct? A particular sequence that you choose. So you can put little SNPs. Imagine this is a chromosome, and imagine in individual 1, you have a sequence that goes like this. Imagine this is a position of a SNP. Here is another position of a SNP. Here is a fourth position of a SNP. Here is a fourth, fifth, sixth, seventh, eighth, ninth. You also have other SNPs here, the ones in red. But let's say you're not going to analyze the ones in red. You're only analyzing the SNPs in black that I've marked as 1, 2, 3, 4, 5, 6, 7, 8, 9. In individual 1, the sequence of this SNP goes AAA. In individual 2, the sequence is ATA. So this A to P is a SNP. Similarly, at this position here, in individual 1, you have a sequence that goes CCC. Individual 2, the sequence goes CGC. So that's a SNP. And so you analyze all these SNPs. You can analyze them by PCR. You have 9 SNPs. You can do 9 SNPs by PCR. Different primers for those 9 locations. And you will determine and get a picture of the SNPs across that entire thing. At this position, this individual has A, C, A, G, G, T, A, T, and C. 9 different locations. Individual 2 has sequences that goes T, G, A, T, G, T, G, A, G. So those are the 9 SNPs. So when you determine the sequences of these 9 SNPs on that one chromosome, you can compare it to the second individual. And you can see the differences that are present in the two at those SNPs. Now, you can do this individually for 9 loci with PCR, no problems. But let's imagine you're going to extend it to all 23 chromosomes, and it's not 9 locations on the chromosome, but you're talking about 900 locations on each chromosome. Now it gets difficult to do. So how do we do it? We use arrays, microarrays. So what you're going to do is we're going to put each possible SNP sequence on the microarray. We're going to isolate genomic DNA from one individual. We're going to label that genomic DNA with a fluorescent dye. We're going to hybridize it to all of the SNPs that are on the microarray. Potential SNPs on the microarray. And see where they hybridize. If they have a perfect match, they will hybridize. If there is no perfect match, they will fall off. That's the trick. So what is a microarray? Let's imagine here is SNP1 with a particular sequence and a change. We have 2 sequences. One possibility could be an A here and a G here. It's the same position on the chromosome, but the sequences are slightly different. Something over here and a simple change right there. So instead of an A, you have a G. Totally different locus, SNP2, different position on the chromosome. Again, you have a sequence that goes GGG, and a different sequence right there that goes a C instead of a G. That's the SNP. Different location on the chromosome. So, what you do is you design oligonucleotides, or little primers, that match this sequence. And match these sequences, and put them in 2 different spots on the slide. So you put each spot corresponds to a particular sequence polymorphism. You design these primers and you lay them out on the dots. Then you isolate genomic DNA from an individual. You label the genomic DNA with a fluorescent dye. And then you add it to the microbe and you let it hybridize. And then you analyze the results. Do they hybridize or do they not hybridize? The genomic DNA, if it hybridizes, it means it's a perfect match. It means it matches to this one sequence, but not this sequence, or it matches to this sequence, but not this. And so based on that, you're able to tell which SNP is present at that location. And because in a little microarray, you can spot 1,000,000 different locations, 1,000,000 different dots. You can analyze all 1,000,000 SNPs at the same time. I'm going to show you this picture that is from your textbook. Here is a SNP. The sequence goes TGGAC. That's on the microarray. The alternative sequence is TGCAC. So here are your 2 SNPs. The SNP is basically this G or C. That's the difference. So, what you do is you take total genomic DNA from an individual. isolate the DNA, cut it up into small pieces, label it with a fluorescent dye. And let's imagine that this individual is homozygous for the sequence ACCTG. He's got 2 copies of chromosomes. Both copies have the sequence ACCTG in this region. So this DNA is labeled. There it is. It comes in, you add the genomic DNA to the slide. It comes in with base pairs. ACCTG, AC, no base pair, TG. Correct? Because this little spot here, there is the SNP, the G to the C transition. Correct? So it doesn't base pair. Now, you raise the temperature a little. And what happens is, because this thing is perfectly base pair, it falls off. The DNA falls off this dot, but it doesn't fall off this dot. And so when you look at them under the light, fluorescent labeling, you find that there is a green fluorescent dot over here, but there is no dot here. And based on that, you'll be able to tell that this individual has the sequence ACCTG at this location. It doesn't have the sequence ACGTG, because that dot is there. And so that's the principle that you use. I'm going to take you through one more. Here are your different SNPs. Let's focus on the first 2 only, 1 and 2. With a particular sequence, we've got a dot on the microarray that picks up one SNP or the other SNP. And then there's this SNP location here, we've got one SNP or the other SNP. Let's imagine that at locus 1, the sequence is TT, homozygous TT. At locus 2, right here, we have the sequence GG in this individual. When you hybridize it, this DNA here will hybridize to this on the spot on the microarray. This DNA here will hybridize to this sequence on the microarray. And so you'll be able to tell that because there's only one spot here and one spot here, this individual is homozygous T for the SNP, homozygous G for the SNP. This individual is basically has the A sequence here, homozygous A at location 1, and homozygous CC at location 2. When you do the microarray experiment, the dot that will light up will be this one because of this match, and this one here at location 2 because of this match. This individual is heterozygous at locus 1, A and P, and homozygous GG. So on the microarray, you're going to get the matching to the A and the T SNP, and here you get the C. And so you can map them in this fashion. And you can of course map all million SNPs. You can put a million different dots on a slide very easily, and you'll be able to map all of the SNPs at the same time using this one genome. Is this kind of clear? I don't expect you to do this experiment, but I want you to understand how it's done. Okay, and so once you've identified the genotype pattern of the entire genome, you can write it all out. Position 1 of the SNP, or SNP1, locus, chromosome 1, SNP1, in individual 1, the sequence is an A. Chromosome 1, SNP2, in individual 1, the sequence is a G. And so on for chromosome SNP3 and for SNP4 and for SNP1 million. You write out the entire variant genotype based on the SNPs, and that's known as a haplotype of that individual. And that's a unique haplotype for that one individual. It will be different from the next individual to the third individual to the fourth. And that's known as a haplotype. Any questions? Should we take a 5-minute break and continue? Okay, let's do that.
Kind of the beginning.